This segment of the Onion Videograph series is entitled Disease Management of Purple Blotch and Botrytis. The video will review common practices used to produce transplanted and seeded onions, especially in relation to disease outbreaks and their management under varied conditions. Appropriate research results from various universities and industry programs will be reviewed. The major objective of this videograph is to aid in the accurate and early diagnosis of these onion diseases and to implement timely management approaches to produce an economical, nutritious, and healthy product for the consumer. Purple blotch is caused by the fungal pathogen Alternaria pori, while botrytis diseases are caused by various species of botrytis. Let us review the range of symptoms and signs associated with outbreaks of these pathogens. Initial purple blotch lesions are small, oval, brown to purple, a ½ inch or less in diameter on leaves. These lesions gradually enlarge and become more elongated and darker in color. Older lesions assume a more concentric appearance and may exceed 2 inches in length. Numerous lesions of various sizes can affect many or all leaves of a plant or group of plants. Older lesions may have a dark gray center as spores of the fungus develop during periods of high moisture. Uninfected portions of leaves may turn yellow, brown, and die as lesions girdle the lower portion of each leaf. Lesions may develop near the neck and actually penetrate the bulb in the field or in storage, producing a brown to reddish discoloration of inner bulb scales. Severe infection may cause more than 75% defoliation before maturity. The botrytis pathogen initially produces small, oval, sunken, whitish spots, usually less than an 1/8 inch in length. Eventually, leaf tips are killed or blasted. Botrytis can also cause a superficial blemish of outer leaf scales called brown stain, or infect seed stalks and cause scape blight, which can be serious for onion seed producers. Lesions may also develop on outer fleshy scales of transplanted and seeded onions at the soil line. Botrytis continues to infect bulbs as they are transported to storage, and there causes a rotting of necks and other parts of bulbs that have been bruised or damaged. The decay is gray to brown, and black sclerotia of the fungus may form on and within infected tissues. Disease development of purple blotch and botrytis is favored by the extent of last season's epidemics and potential carryover of infected onion tissue and bulbs. Improper disposal of onion culls can add to this season's disease pressure. Lack of a proper crop rotation can also add to the season's disease pressure. Continual cropping to onions aggravates disease losses. Damage to leaves by thrips can predispose this tissue to colonization and infection. Likewise, storm damage can also provide entry points for these pathogens. Cultural practices which emphasize excess and late-season applications of nitrogen fertilizer can delay crop maturity, increase canopy density, and predispose onions to infection. Environmental conditions which favor sporulation and infection by purple blotch include periods of low to moderate temperature and high moisture. Likewise, botrytis is favored by slightly cooler temperatures and high moisture. Both fungi produce spores on previously infected tissue and are blown from these infected sources to new plants where infection can occur during periods of favorable weather. Environmental conditions can be monitored during the season in relation to crop development and the potential for disease outbreaks. Daily access to this weather information can be improved by the use of satellite or cellular telephone communications. Knowledge of weather patterns and the potential for disease outbreaks can help plan scouting routines and schedules to detect potential problems before they become uncontrollable. The stage of plant development when foliage damage or infection occurs is related to loss in yield and quality. This is especially important during the last 8 weeks of crop development, at a time when purple blotch and botrytis often appear. Proper sanitation is important to remove previously infected or contaminated sources of plant debris and culls, which can provide this season's disease pressure. Fungal pathogens can grow on this tissue, and spores are then blown by wind to new plants to repeat the disease cycle. Crop rotation and crop alternatives help reduce disease pressure. Onions should only be grown once every 3 to 5 years in a field. Rotate with other vegetable crops such as potatoes, lettuce, sweet corn, cabbage, cauliflower, and broccoli. Legumes such as dry edible beans, soybeans, and alfalfa are good components that help reduce the carryover of plant pathogens between crops of onions. Small grains such as barley, oats, and wheat, in addition to field corn, are also commonly used in onion rotations. To produce a high-quality crop of onions, it's important to select the appropriate variety for a specific production area. Avoid late-maturing, heavy-topped, and thick-necked varieties. Follow local recommendations for planting dates, good seedbed preparation, and the use of certified seed. Cultural practices should emphasize timely cultivations that minimize plant damage. Avoid late-season applications of nitrogen. When environmental and disease threats exist, timely applications of appropriate fungicides can reduce damage and secondary spread of fungal pathogens. Fumigation should use the smallest amount of water possible, ideally less than 1/4 inch per acre. On the other hand, aerial application should use the largest amount of water possible, ideally 5 gallons or more per acre. Ground rig-based systems often use 25 to 50 gallons of water per acre, and the fungicide can more successfully penetrate the plant canopy to reach onion necks and tops of exposed bulbs. Regardless of application method, early detection of a disease threat followed by the thorough application of an appropriate fungicide on an effective schedule can increase yield and reduce quality losses from purple blotch and botrytis.